Hello again. So my name is Michael Kearney, and you would have seen a previous lecture from me about the concept of a mechanistic niche model. What I'm going to do in this lecture is tell you about a software package called NicheMapR, and this software allows you to do that integration between biophysical ecology and dynamic energy budget theory. So what I'm going to do more specifically is introduce you to the NicheMapR package, where it came from, and generally what it does. And then I'm going to show you a series of web apps that you can play with that basically give you a head start into seeing what the NicheMapR package is able to do. So you can just play with those apps and then get a feel for it, and then you can actually go in and use NicheMapR in R. And then finally, there are some practical exercises that I've produced for the course, and I'm going to go through those practical exercises that give you a hands-on experience using NicheMapR. So first of all, where did it all come from and what's in NicheMapR? So as I mentioned in the lecture I gave previously, there's this field called biophysical ecology, which was developed by Warren Porter and David Gates, and this was the application of heat transfer principles to organisms, actually, in general. And so Warren Porter and his colleagues, who really pioneered this, they developed some software to make the calculations. And this was back in the late 60s, early 70s. And in those days, you programmed in Fortran. And in fact, Warren was working with punch cards back then. And so, really early days of programming. Fortran is a really powerful, fast language for computation, but a little bit tricky to work with, especially when it was in punch cards. But even still, these days, compared to programming in Python or R, it's much harder programming in Fortran. But that's where the original models were developed. So this is the scheme of the microclimate model that comes with the NicheMapR package. So Warren and his colleagues realized they needed a sophisticated model of the microclimate to solve the heat budget equations that they were interested in. And this is the scheme of the microclimate model with a whole lot of different Fortran routines that are doing things like working out the solar radiation from first principles, interpolating daily maximum minimum temperatures and humidities and so on into hourly, if that's the information you have, working out properties of air when the air is moist or dry, working out soil properties, and then doing a big calculation of the heat budget of the soil, accounting for snow and also for soil moisture. So all of this is going on in Fortran in NicheMapR, and we originally called it NicheMapR for reasons that should be clear from my previous lecture. But what I've done is I've incorporated this into the R programming environment. So it's what's called NicheMapR. So what happens is that whole scheme is just called by a wrapper in R, and R acts as the way of getting the information that the model needs in, and then giving it back out to R so that you can actually do things in R with the output. But all of the hardcore computing is happening in Fortran still, and that makes it super fast because it is doing a lot of calculations, but allows you to use the power of R to actually bring together data that you need to run the models, and also, of course, the graph. It's much easier to make graphs and that sort of thing. So the Fortran wrapper is called by a series of, in terms of the microclimate model, a series of different scripts that bring together data sets from different sources. So there's one called microglobal, for instance, and so that takes in input from the user. So you might put in your location, you might put in what the slope and aspect is, what the shade level is, the soil type, into microglobal, into that function. And that function will then get the required information to run the model from a number of databases. So it will get the data from a global climate database that's developed by the CRU in the UK, which is a global climatology of monthly mean conditions, max min values for the typical day of each month, for all the things that the model needs: temperature, cloud cover, humidity, wind speed. It also gets soil moisture information from a soil moisture database. If you're not, the model has a soil moisture routine, but if you don't run the soil moisture routine, it gets pre-computed soil moistures from this database. And then it also gets soil properties from some data that's in the form of a table from Campbell and Norman's excellent book on environmental biophysics. So it's drawing on these particular resources in terms of the climate and soil type. And the new et al. Paper describes this global climate data set. So it's about I think 11-kilometer resolution boxes across the whole world for the typical day of each month of the year. And the soil moisture similarly. Also, the solar radiation module within the NicheMapR package within the microclimate model is going basically from the solar constant, how much solar radiation is hitting the outer atmosphere, and working out how that is attenuated by the atmosphere itself according to the angle of the sun. And it's quite sophisticated, but it does need information on the aerosols in the atmosphere, which are having a bigger impact on the overall solar radiation level. So there's a global aerosol data set that's being called by the model as well. That's at a relatively coarse resolution, and so yeah, you might see some blockiness in the outputs as you go from one pixel on this to another. But it is accounting for the dirtiness of the atmosphere. And I certainly noticed when I was running these models for Australia compared, well, initially before bringing this aerosol data set in, I was getting underestimations of the solar radiation for Australia, and ultimately worked out that it's because the model was set up for the US, where there was a lot more pollution in the atmosphere at the location that was used to estimate aerosol properties. And Australia has relatively clear skies in terms of pollution and made about a 10% difference in the solar radiation. So that's going in as well. And then this microclimate model then produces tables of output that you can then use that include above ground conditions in terms of air temperature, wind speed, humidity, solar radiation. That's the met out table. And then soil temperatures at different depths for each hour of the day, as well as soil moisture, soil water potential, and humidity. So all of these details you can find in the little tutorials that I've made, including the things that I've made for this course. So I won't go into all the details here, but just to give you the sense of what's going on when you run the NicheMapR microclimate model. So there's one that runs this global climatology for the typical day in each month, but there are other databases, including a global historical database that allows you to go anywhere in the world back to about 1957 and actually get historical conditions. There's one specifically for Australia, one specifically for the US, one specifically for the UK, one specifically for New Zealand as well. So that's the microclimate model, and then there's the ectotherm model, which actually I should have mentioned previously. There was a paper describing the microclimate model in ecography that's got all the gory details about exactly how that works. And then there's a paper from last year, from 2020, on the ectotherm model, which also incorporates the dynamic energy budget model, as you'll see. So the output from the microclimate model goes into the ectotherm model, which is basically solving those coupled equations of heat, water, and nutritional balances in this function called ectotherm.r. And that produces outputs that include, for each hour, what the environment that the animal experiences is, including its body temperature, but also what was the actual air temperature where the animal was, what was the wind speed, what was the humidity, and so on, accounting for the animal behaving, moving underground and changing how much shade it uses and so on in its habitat. It also has a table of all the energy balances, so you can get the fluxes of heat coming in and from solar radiation, or the heat being lost from infrared, all those terms that I showed you in the heat budget equation in the previous lecture. You can get the actual values of this per hour. And then you can also get the mass balance, aspects of the mass balance, including, if you're not running the dynamic energy budget model, it will give you the water loss in terms of respiratory and cutaneous water loss, and it will give you an allometric prediction of metabolic rate and therefore oxygen flux. but if you run the DEB model, it gives you all the DEB model mass balance outputs. And then it allows you to make characterizations of the suitability of that site for the organism, and for instance, make a plot of the activity window, which I'll explain in a little bit more detail in a sec. So the ectotherm model has a lot going on inside it as well, as described in this paper. So the ectotherm model is taking in the microclimatic conditions from the microclimate model, and it does this for 2 different levels of shade, and they are the options that the organism has to choose between. So it's a spatially implicit model in terms of the shade, but also all the functional traits that the model requires. And so they relate to morphology, behavior, and physiology, as well as the DEB parameters if you're running the DEB model. And they all go into the controlling Fortran program of the ectotherm model, which then does calculations of the geometric characteristics of the organism, its surface areas, and so on. It also has an algorithm for computing the steady state body temperature, which is of course then drawing from all the algorithms or subroutines that are computing things like the conduction, heat exchange, the radiation out, the metabolic rate, the respiration rate, evaporation, conduction, solar radiation, and infrared coming in. So all the terms of the heat budget, and all the time going backwards and forwards, getting the properties of air in terms of the properties of wet air and dry air that it needs for all these calculations. And then it's got some sophisticated algorithms for behaviorally thermoregulating to figure out where in the environment the organism should be. And then back to R comes a table which has, as we just saw before, the environment, the energy balance, the mass balance, but also if you're running the DEB model, it's got a table of the DEB model outputs as well, and yearly summaries of what's been happening to the organism, especially if you're running the DEB model. Now, I haven't really talked about the endotherm model, but there is one. It's actually mostly finished. It's sitting there in the NicheMapR package. There is a software note that's currently in review that's almost finished. And this is basically solving a heat budget for an endotherm, and basically using similar principles to what I described for an ectotherm, except you've got the body temperature as a known entity, and you're trying to find the metabolic rate and water loss rate that balances the heat budget equations. But there's a lot of complexity and different details involved in endotherm thermoregulation that this model handles. So that will all be described in that software note, and there are also tutorials and things in the package explaining it in detail as well. And then, yeah, the ectotherm model, this is the scheme I showed you before. If you turn the DEB model on, there are a series of routines that are also written in Fortran that are running the DEB model. And so each hour, all the heat exchange and water exchange is computed, and then it calls the DEB model, gets the size for the next hour and onward. And so this DEB scheme here has the normal standard DEB model that can also handle metabolic acceleration. But it also has a holometabolous model that's a far more complex model because it's doing the pupal stage and quite a different situation in the larval stage to non-holometabolous organisms. So all of that's going on as well in the model if you turn the DEB model on. Also in the NicheMapR package, there is code or functions to run the DEB model as a simulation independent of the heat budget. Just as, in fact, it's essentially taking what's in the Fortran code for simulating a trajectory through time and just providing that for you to play with, which can be really helpful in just understanding how the DEB model is working, and also just seeing how your particular sets of parameters actually translate into a trajectory, which is not always obvious when you're doing the parameter fitting with the add my pet routines. For me, if I had this back in 2002 when I was trying to understand DEB theory, it would have certainly sped things up for me, and a lot of pennies would have dropped faster because you can just see what the DEB theory is doing in terms of predicting a life cycle. So they can be useful for that, and there is a little tutorial that I'll mention that I've made that shows you how to run those. So that's what's in the package really briefly. Now, as I say, there are some apps that I've made that you can play with and just get a feel for what these models are actually doing, especially what the microclimate model's doing, and what the ectotherm model's doing, the sorts of levels of detail that are available in them, and what sort of results you can get, which, you know, it's just fun to do on its own, but you can use it as a starting point to learning how to do it yourself in R, and then obviously you've got far more control if you're running these models in R yourself. So this web page here links to the page you can see on the screen, which is where I've put all of the different apps that are running NicheMapR things that you can choose from. And there's currently 11 of them, and there's a few more that I have planned. But yeah, the first one is a microclimate, runs the microclimate model. This is what it looks like if you go to that website. It's got a picture of Uluru in Australia, and actually it's simulating by default at the start conditions in the centre of Australia at Uluru. Here it is in the little map here. So you can go anywhere in the world with this little map. You can use the little icon here, put it somewhere, and that will run, and then hit run, and it will run the model at that location. As you'll see in a sec, you can zoom in and out of this map. And you can see when you click on that location, what happens is it queries a database of environmental conditions at that site, the new et al. Database that I mentioned. It also queries a function that gets digital elevation models for anywhere in the world. And so you can turn that on or off. Takes a little bit longer when it's on, but you can see this has actually got the fine scale, I think it's about 30-meter resolution elevation at the exact point where I put that marker, so you can see the edge of the rock there. And it runs the microclimate model for that site and shows you at the top here the soil temperatures at different depths. So this plot shows on the y-axis the temperature of the soil, the x-axis the hour of the day, and you can see these different colors at different depths in the soil. Red is right on the surface. You can see the temperature is fluctuating dramatically, goes up to 50 degrees on the typical day in January, and then down to about just under 20 degrees at night. And you can see as you go deeper and deeper, the soil temperatures become damped in their fluctuations and peak later and later in the day. And you can choose which month you're looking at in terms of that soil plot. You can choose which depths you want to see. You can plug the latitude and longitude manually in here if you want. And also you put the height of the the height of interest for the microclimate model here as well. So, well, this is just zoomed out to show you where in Australia we are here in this particular simulation. And then these plots here, you can see if you scroll further down, they show you the air temperature in the dashed blue line through the day, which is what is from the weather station. So basically the maximum and minimum temperature from that data set, that monthly data set, interpolated into an hourly cycle. And then in black, what that temperature would be at 1 centimetre above the ground. So the dashed blue line's at about 1.2 metres above the ground, whereas the black line is at 1 centimetre above the ground. And you can see that it gets colder at night near the ground and also gets much hotter during the day near the ground. Got the same plot for humidity and the same plot for wind speed. Notice the wind speed is much higher at the blue dots, which is what the weather station level of about 1.2 meters says, whereas the 1 centimeter wind speed is much lower. And you can also see the solar radiation in watts per meter squared through the day, sunrise and sunset. And you can see that the solar radiation goes up and then all of a sudden is truncated here. And that's because the rock, Uluru, is blocking the sunlight at that time of the day. So it would be completely in the shade. So that's the sort of level of sophistication that's going on with the microclimate model. If you click on the show terrain and soil parameters, you can then get access to the things, the parameters of the microclimate model, like how much shade, what the slope and aspect is. This is coming from the digital elevation model if you've got that turned on. Also the horizon angles, which is the angle to the sky for where the sun can be, which is in this case being blocked by the rock in certain directions. The solar absorptivity of the soil, also known as the albedo, the emissivity of the soil, which is an important property, the thermal conductivity, heat capacity, and various other things. So you can see that fine scale topography is being checked here, which is using that digital elevation model. And so that's basically determining what the slope, aspect, and horizon angles are. But you can turn that off and control them into any values you want. So there's various options there that you can play with, and you can take this anywhere in the world. So that's with the global monthly climatology, but there's also, as I mentioned, a database. It's actually reanalysis data from the NCEP, National. Oh, actually, I can't remember what NCEP stands for off the top of my head right now, but it's basically a project to reanalyze. It's sort of a combination of model and observed data that gives us all of the things that you need to run the microclimate model. I think National Centers for Environmental Prediction is what it stands for. And it gives us all the things we need to run the microclimate model every 6 hours, going back to about 1957. Oh, actually 1949, I should say, 1949. So you can run it off this database with the Soil NCEP app. So there's the address there. It's also, you can get to it from that landing page I showed you before. And by default, I've put it in Namibia, out in the desert, actually at that exact site that you can see there. And you can see now we've got a plot of the whole year soil temperature. It only runs 1 year. You can start it at the beginning of the year or in the middle of the year, but it will run just 1 year. It does a little burn in of the year before to get everything going, and then shows you 1 year. And so this is for 2019. And you can see the deep soil temperatures again, very stable. And then it's fluctuating dramatically at the surface. And you can see, because it's not the long-term average, it's actually of the month, it's actually daily. You're getting soil surface temperatures really high, up into almost 80 degrees in some places, which actually happens. But as you go deeper into the soil, it drops off very quickly. Note also, you can, if you have observations of soil temperatures in that place, you can actually click on plot. You can actually upload observations, and then you can plot the observations. And if you've got parameters you've saved, you can actually you can save them by clicking on this input, this checkbox, and save the parameters for an analysis you've done, and then you can load them in later as well. So that's the microclimate model. There are a few other ones. There's one that does the US as well, and then there's the ectotherm model. So I haven't updated this, but that should say ectotherm there. So there's an ectotherm app which runs the ectotherm model using the micro global function. So basically the same as what the first app that I showed you. So the monthly long-term average climatology is being used to drive the ectotherm model here. And you've got access to all the microclimate parameters and plots in this app just as before, but it's also showing you the outcome for an ectotherm in terms of, in this top left graph, the body temperature of the animal through the day in January. So this is just plotting, again, the typical day in each month. And it's got, this is the body temperature in black. And you can see that there are these thresholds here that are some of the parameters of the model: the critical thermal minimum and maximum, the preferred maximum and minimum foraging temperatures, as well as the preferred temperature itself. There's also the temperature at which the animal will come out and bask, and also the temperature at which the animal will emerge. So if the temperature where the animal is below the emergence temperature, it's stuck underground, even if the conditions are good on the surface. They will come out and bask if they can be at the basking temperature or above, which may not be suitable for activity, but they might still want to come out because it's warmer out of their retreat than in their retreat. So you can see in this case, the animal was basking for the first couple of hours in the morning and then was active. There was a period where it was inactive in the middle of the day because it was too hot, and then it was active again. It's the orange dots are active, the blue dots, which you might be able to see, basking, and then it retreated back to its burrow. You can see on the right how much shade it needed. It was able to have up to 90% shade in this simulation, and you can see it increasingly uses shade through the day. But then when it's forced to retreat to the burrow because it's too hot, then there's of course no shade because it's in an unshaded burrow, and then it comes back out again later in the afternoon. And this plot here shows you the depth of the animal. So it's at 2.5 centimeters overnight in its burrow. It comes out onto the surface for a few hours, retreats deep underground to get away from the heat, back on the surface, and then back in its burrow. And this plot down the bottom here, you saw something like this in a previous slide, is plotting the activity window. So this is the month of the year and the hour of the day. And this particular line here is basically showing you the same information as this graph up the top for January. It's saying in dark blue when it's nighttime. So that's till about 5 o'clock. So there's this period here, sunrise is here. And then it actually basks for the first couple of hours. It's suitable for basking. So that's these two points here. And then it's active for a while. And then the white is inactivity because it's too hot, and then comes out again, and then into its retreat at night because it's a diurnal animal and this is nighttime. But you can see now also across the whole year, each month, how many hours it was active and when. And it sums the whole lot for the year at the top here. So 2,731 hours for this animal at this location. And the parameters, when you click on the show ectotherm parameters, include the mass of the animal, the height above the ground, and of course big animals are higher above the ground, the shape of the animal, which can be a lizard shape or a frog shape, but there's also a typical, like there's an ellipsoid shape and a cylinder as well. You can stretch the shape out as well. So you can change, you can have a more spherical or a more elongate sort of ellipse shape if you've chosen the ellipse by using this stretch thing. You can choose whether they're diurnal, nocturnal, crepuscular, or any combination of those together, whether they're allowed to seek shade or go in a burrow. If they go in a burrow, whether that burrow is in the shade. Also, they may have the option of climbing if that's something they will do, because a lot of animals will climb to get out of the heat. You can say what the minimum and maximum depth the animal can go underground to are. So you can make it so they've only got very shallow burrows if you want to see the consequences of that. And then here are all the parameters relating to the thermal behavior. So thermoregulatory behavior. The minimum foraging temperature set here at 30 degrees and 35 to max. The preferred temperatures down here in the middle at 33. They'll bask as low as 20 degrees, but they won't start moving until it's 30. That's their body temperature, of course. The temperature at which they leave the retreat is set to 10. The critical thermal minimum and maximum 5 and 40 respectively, which are determining how deep the animal goes in its retreat. And you can say what solar reflectance the animal has. Here it's absorbing 85% of the solar, but that could be changed. How wet the skin is. And then there's even a way of constraining whether the animal comes out according to whether there's a signal of warmth coming from, if it's in its burrow, coming from into the soil. So quite a few different things you can play with there. And again, this should say ectotherm NCEP. This is the NCEP version of the ectotherm model that runs the ectotherm model from the historical data. This is the same. This is the ectotherm model run for 2019 in Namibia. So you can now see the body temperatures across the whole year, and you can see the activity window across the year, the shade selected and the depth selected. So you can see in Namibia, this animal could be active throughout the year, though there are windows in the summer where it's too hot to forage. It's a bit hard to see that because we're plotting a whole year, but if you choose here which month to plot, you can zoom in on those months and see it in more detail. As I said before, like if you go to save input output code, you can of course save your parameters, but you can also get the output of all these models as a table, CSV file. And also you can get the code to run the exact simulation that you just ran in R. So we'll just give you an R script and you can take that into R, and if you've installed NicheMapR properly, it will just run that simulation so you can see exactly how to get these predictions from that you got from the app in R yourself, and then start modifying it from there to your purposes. And finally, this is a bit new, very new actually. This is an app that runs simulations of the DEB model, and it's basically using the DEB functions that are in NicheMapR that I mentioned that allow you to just run the DEB model simulation to get trajectories independent of the biophysical models. But yeah, this app has access to all of the species that are in the Add My Pet collection, and you can type in a species up the top here. It will auto-complete. So you can't, when you click on the drop-down menu, you won't see all 2,941 species that are currently in there, but you will you'll see some of them. But if you start typing, it will start to find that species. And then by default, it's running Daphnia magna, which is of course the species that started DEB theory off. So there's some pictures of Daphnia magna up the top. And it's plotting the wet mass as a function of time, showing you the partitioning of the wet mass into structure in green, reserve in grey. It's got a stomach model, so it shows you the food in the stomach in brown, and then in pink, the reproduction buffer, which is bouncing up and down as the animal has its eggs. And then it's out of interest, it's plotting the oxygen consumption rate as a function of mass, showing you that in this particular case you get 3/4 power scaling of metabolic rate with size through ontogeny, but that's not always the case, of course. And then you've got these plots down the bottom, which are showing the partitioning of the various powers, the various fluxes of energy to the different processes in the DEB theory: assimilation in grey, reproduction in red, maturation in green, growth in yellow, and maintenance in blue. And the life cycle you can see marked by these dashed lines: birth and puberty. So you can control some aspects of the simulation, not everything, with the interface here. So you can set the number of days it runs for. You can choose an hourly or a daily time step. If I chose a daily one, these are actually plotted as vertical lines. So if I chose the daily time step here, you'd just see a whole lot of vertical lines spaced out. But because it's hourly, there's enough lines that it looks solid, just so you know. You can change the temperature. You can change the F, so as if controlling food restriction. You can start the simulation off at different stages of the life cycle. You can tell it what the clutch size is. You can tell it what units to plot the results in. And I've also put in a slider so that you can change the scaling. Well, you can change the size of the animal according to the DEB covariation rules. And because everyone's always interested in kappa, especially when they're learning DEB theory and wondering how does kappa work, you can change the kappa by multiplying the current value of kappa. And this will come a bit later. It's not in there right now, but I'm playing around with a way of generating the temperature size rule from DEB theory that involves this slider, but I'll put that in when I finalize that. That really wasn't supposed to be there. You will see a series of plots of the mass fluxes, oxygen consumption, carbon dioxide production, metabolic water production, nitrogenous waste, dry food intake, and wet feces output, all as a function of time. And you can get the output by clicking on the Save Output and get the CSV of the of that simulation. So it's a really good way to just get a feel for the DEB theory and also to see whether the trajectories look realistic or not. Okay, so those are the apps, and they're sort of ways, sort of inroads or gateways into playing around with the models if you're unfamiliar with them and just want to get a feel for them, especially if you're wondering how much time to invest learning them. Obviously, you're investing time in learning the DEB theory doing this course. This will also give you a sense of whether you want to invest time learning the background behind the biophysical models, because you really need to know the background to use them properly, just like with DEB theory. You don't want to just play around with these things only vaguely understanding the underlying theory. But this is a good way, those apps are a good way to get a sense of whether you can be bothered spending all that time learning the theory so that you can properly use the models. And so if you do want to go further, I've got these little practical exercises that I'll provide as PDFs and our markdown files for you to play with. And I'm just really going to briefly show you what's in them rather than going into all the details, and you can just go through them at your leisure. And hopefully there's enough description in there to get across the points. So there are three documents that I provided, sort of part one, NicheMapR and DEB theory. And part one is looking at the microclimate and ectotherm models on their own. So this has instructions for installing NicheMapR. And then it shows you how to run the microclimate model using that global data set, make some plots. Then it shows you how to run the microclimate model with the historical data set. And then it shows you a simple calculation of, or shows you a calculation of the heat budget, how to run the heat budget model with the ectotherm model, but with the DEB model turned off. And so you'll make these plots of microclimatic conditions, soil temperatures, and this is with the NCEP data, so more detailed, daily, historical. Same with soil temperature. So these are all the plots that are generated by those scripts. And then this is the heat budget model output for the ectotherm model, plotted a different way to the app, where it's showing everything in one hit, body temperature in black, the shade selected in green, the activity in orange, and the depth in brown, but it just shows you how to run that particular simulation. And then it'll make this activity window plot. So it's for a sand lizard in Halifax. So at this time of year, it shows you that the sand lizard would be well and truly in its activity window if we were all together in Halifax and sand lizards occurred in Halifax. Then there's a second one that actually shows you the DEB functions within NicheMapR. So it shows you how to read the allstat.mat file into R. It takes a long time, but you can bring it, you can turn it into an R object, and then you can use that instead of the allstat.mat file, and it loads much more quickly. You can then, it shows you a few examples of playing with the allstat.mat file in R to get information out that you might be interested in. And then it shows you how to run a DEB simulation using the DEB routines independent of the heat budget. And then it shows you what happens when you turn the DEB model on within the ectotherm model of NicheMapR. So here's an example from the part which is playing with the allstat.mat file, where I've pulled out all the lizard metabolic rates and plotted the log DEB modeled oxygen consumption rate as a function of mass. And this is an example plot that is produced by one of the DEB functions in the package. And then this is a plot you'll get when you run the DEB model for a sand lizard as an example under realistic conditions using the NCEP data. And actually you'll see that you should be able to get that model to look like this. The way that it's set up in the initially, the animal doesn't do too well over winter, and there's a little challenge for you to work out what parameter to change to get the graph to look like this, where they're actually much happier over winter and not losing a whole lot of condition. And it has something to do with the temperature correction. And temperature correction is really important, of course. And in DEB theory, we're using the Arrhenius temperature correction, and we need to specify all the rates that we have at a particular, we need to say what temperature they happened under. Now, one challenge you'll frequently encounter is that the animal wasn't at a constant temperature. So you need to somehow give it a temperature for the univariate, zero variate data, even though it might have been under fluctuating temperatures, because a lot of animals can't actually be kept at constant temperature in the lab, for instance. You might have lab data on reproduction rate, but it's actually not constant temperature. But there is a way to convert fluctuating temperatures into a constant temperature equivalent that you can then put into the estimation routine. And similarly, often you might have observations of, say, reproductive rate in the wild of an animal that's living in a terrestrial environment, its body temperature is totally different to air temperature. So what you want to do is know what was the appropriate temperature correction factor to put into my observation of field reproductive rate or something like that, or field development rate. So there's a final third PDF that explains how to do these temperature corrections, so that you can take a sequence of variable air temperatures, for instance, convert them into the actual temperature correction factor. This is the multiplier on the reference rate. So at 20 degrees, it's 1. And then these are situations below 20 degrees where it's going down, and above 20 degrees where it's going up, but non-linearly because of the nature of the Arrhenius equation. So you get these correction factors up to about 3 in the hottest for this particular example, and down to near 0 in the coldest. The same with this is running something in the field where you're actually accounting for thermoregulation. So this is the body temperature of the animal through time, and this is the correction factor for the animal through time when it's thermoregulating, and you can see that it's spending a lot more time with much higher correction factors, so the rates are going much faster when you account for thermoregulation. So you'll see all that in that script, and it will show you how to go from a mean. You basically can take a mean temperature correction factor for this for the period of interest, and then turn that back into a temperature that's equivalent to the mean constant temperature, so the mean correction factor. So that's the notion of a constant temperature equivalent. It's explained more clearly and more slowly in the little document. So this is an important trick to know for estimating DEB parameters and getting the temperature in there correctly. That's the document. So it talks about installing packages, how to get NCEP data for air temperature at a particular location and time, and then how to compute the constant temperature equivalent based on that air temperature. And then it's also showing you how to use NicheMapR to estimate body temperature and include thermoregulatory behavior so that you can get the field constant temperature equivalent if your observations are for field conditions at a given location. Okay, and finally, some resources for NicheMapR. First of all, there's a Google group for NicheMapR. So this is the address up here. So you just have to request membership and I'll let you in. And then there's currently about 130 people in there. And I just prefer any questions about NicheMapR to go in there so that everyone benefits from the question and the answer. And you can answer each other's questions and I'll answer questions. And I try and really answer questions quickly in that forum to encourage its use. So there's that. Of course, there's also the Twitter handle for NicheMapR, and this is my Twitter handle as well. So I actually make most announcements about NicheMapR on those Twitter handles. NicheMapR is all open source and on GitHub. So this is the GitHub repository for NicheMapR. And also I've made a website that's basically an overview of the project, of NicheMapR itself. Lots of information about the background, the people involved, the models, all the vignettes that are part of the package are also visible there under help, news and related projects. So there's a few resources if you're interested in using NicheMapR that you should have a look at. And thanks very much for listening. And I should be able to answer questions you have, maybe live, maybe indirectly.